Thank you for everyone for joining the meeting today. So today we have a really exciting webinar. Paul Shan, our CEO, he's going to be going through the new features in Peak Studio 13, as well as our new software, ProteoformX. So we're really excited about these two releases that we just had. So Paul, I can hand things over to you and you can get started.
First, thanks, Jenny, and for the introduction. And I think, today, I will take the following time, like roughly 45 minutes, to introduce our new release. And first, I would like to thank you all for your time and also your interest for our product. And before I introduce the product, I first thanks all our team members, and they put much effort to develop this product as we have done in the past years. Our first this release for Pixel, we are main changes or main functions is focused on DI data analysis and try to improve the speed and the sensitivity and accuracy. And for particular application, we are focused on explore DI capability for uncover the lower abundance peptides, especially those sequence variants and the novel peptides by integrating database search and de novo sequencing. And another one is we considering currently in the community that we have a bigger concern about FDR estimation for DI data. Most of the engine software are black box. We try to provide a detailed insights of the data with the result for a researcher to validate, to view, such that they are confident about the result. Surely we will continue to improve discovery and target the proteomics. And considering now dark proteome or peptome are getting more and more attention, and we continue to enhance our peptome workflow, the peptide is the main focus. Protein or other information used as the source of the peptide will come from. So more specifically, I will continue and before we talk about the detail about the algorithm functions, I would like to take some time to recall PIC's design logic. So, and to my knowledge, the general proteomics workflow has some processing, mass spectrometry, and data analysis. And PIC's team believe and understand data analysis is not just a tool for get the result, actually is a part of the whole workflow. And in considering this means from the workflow, we get sample and we mass spec. Finally, the output is a raw data file. There it contains all the information regarding sample processing, mass spectrometry method, and if, for example, either it's DDA or DIA, or whether it contains ion mobility, whether use which enzyme, whether use which instrument, for example, whether use Astro or timsTOF HT or ZeroTOF 8600, and each instrument have, and whether your sample is well known, they have very good database information, or your like research topic is some say they don't have enough proteome information. All those one is contained in the data. So picks we are based on those ones, started from algorithm, started from detect precursors, which is generated by some peptide. Then if we have tandem spectra for that precursor, like DDA acquired, or you have some chimeric spectra, even you didn't know, but that spectra contain that information, we will do association. And Finally, we are based on those one to identification and quantification. Very often, we will for identification we use different approaches, de novo database, library search, and for quantification we can use chromatogram, or we can also use Apex information to get the quantitative information best fit with the data. So this is a logic. So what you can expect from PICS algorithm, and you can see it's a peptide precursor feature-based solution. What does that mean? And so let's say from whatever the sample from, it's a mixture of protein. And from there, we can use different enzyme. Let's say typically we use trypsin digest. And then what we get is a tryptic digest. And then we load to the column and use mass spec acquired data. In the data, it contains all the peptide precursor information, which is from those tryptic digest and get elute from the column, ionized in the mass spec, acquired in our mass spectrometer, and recorded in the raw data file. Now, for pigs, when we get those raw data file and we set the parameter, we first algorithmically detect the peptide precursor, like here. You can see the first step is detect those precursor feature. From the feature, and then we know the instrument will tell us if there is a tandem spectra targeted to this precursor, and we will associate. If there is a co-eluted precursor, and the chimerical spectra algorithm will take care of those. The software will assign this chimerical precursor associated with this spectra. Finally, we get the identification of those peptides, and we will summarize all the peptides and use parsimony algorithm to infer the protein. Finally, you get a protein list, which shows the coverage by supporting peptides. And more than that, because our search engine can integrate de novo database information. For some peptide, they don't match exactly with the sequence. The de novo has a very good sequence tag associated with this protein sequence. We will list there as a gray bar. And by doing this, and we present the result. So detailed information about how to do identification, and say given a tandem spectra, the peptide sequence is derived from a tandem spectrum. Regarding the algorithm, and there are many, they can say, methods. Technically, they are spectra-based or peptide-centric. As for spectra-based centric means given a spectrum and try to find a peptide you believe is a good match. And for peptide-centric is given a peptide, goes into the spectrum, find the evidence for this peptide. So it's just based on the starting point, we have peptide-centric and spectra-centric. And in pigs, so if you give me the peptide, for example, library, we will check for each peptide in the library whether there are evidence, whether those spectrum match with this. Or you give me a sequence database, and then we will use spectra focus. Given the for each spectrum, can we find a peptide in the database which best match this spectrum? Or we can directly use de novo, given the spectrum, come up with a sequence which best match with this spectrum. Those are three most common methodologies, and with DDA and DIA. Besides those three, PICS also have another one we call homologous search. The homologous search was designed for, okay, with a de novo sequencing. and a database, then in this case, can we find a better sequence considering both de novo and a database? What this function can bring to us is if your database is not very complete, for example, some Asian species, and we only have partially sequence information, or some dark protein, non-coding regions, or then your de novo sequence you need a perfect spectrum, you need a very good spectrum. In this case, your de novo sequence is not very good, but you get some sequence tag with some ambiguity. By combining both of those, you get a better sequence. And for the application, what we get after PIX identification, you can get the library peptides, database peptides, modified peptides, sequence variants, or novel peptides. And this is our identification. And in terms of this, we will organize our result in a way for you to either validate and link the result with raw data. Start with the feature tag for each peptide precursor, regardless it has ID or not ID. We give their MS, like say, heat map information and their chromatogram information. If there is a tandem spectrum associated, like in this case, and they have ID, we will show the spec. And if this peptide is come from protein database, and you will have protein information associated where it located this supporting peptide. And if you want to check raw data, okay, you can go MS2 information. In this case, you can see this is a chimeric spectrum. The instrument target is a higher abundant. However, there is a chimeric precursor also co-united. Software will give this as a primary and secondary and chimeric spectrum. And if we want to see details about all one from LC-MS view, it will show you all detected precursor and with the tandem spectrum we acquired. For those tandem spectra, they may have database peptide, they may have de novo peptides. We list all of those. By doing this, and we give deep insight about of the data. And same as DIA, for example, you given any precursor we detect, and either this is database peptides or variants or novel peptide. Then from those peptides, we infer protein and link to the protein information. And here you can see this red box circled is the linkage between this peptide to the protein to the raw data. And for either validation, for each precursor or peptide we detect, we will give the precursor information in the raw data. Also for DI, we give them fragment ion chromatogram information. And for quantitative, and we give like their chromatogram and apex information presented in a single view. With this one, you have a full insight about data and the result. And here is one example of a protein we identified from DIA data. You can see this is a protein, and with this protein, the blue bar support there is a precursor of peptide matched with this sequence. For example, like this one, obviously is this DIA spectrum. The other peak may come from precursor in that window. And also you can see with this coverage, there are peptide variants, even some novel peptide with a good sequence tag associated with this, like this gray bar showed there is a precursor. The sequence is not exactly matched to this one. However, by de novo, their sequence contain a tag started from here matched to this. This will shed some light for the relation of this peptide to this protein. So this is the result view. And it sounds very complicated or very sophisticated algorithm. However, we design those algorithm and arrange it based on the application. For each of those one, we provide a one-button function or workflow. Specifically, we have proteoform workflow and peptome workflow. For proteoform workflow, there we try to answer the question, given a data set generated from some biological sample, what are the protein in the data? How much of those protein? We try to answer this question. Yeah. On the other hand, the peptome workflow is given a data set. What are the peptides present in this data set? The protein or other source information is trying to tell us where this peptide come from. If they exactly match the sequence, it could be this is a database peptide. If it's a sequence variant, or we know it's a sequence variant, or it's a new peptide, they share some tag with the protein or a few of proteins. For example, one peptide, their N-terminal segment may share with some part of the protein. The C-terminal may share with another part of some other protein. This more likely give us a hint. This peptide might be a fusion peptide, come from either splicing of the same gene or fusion of different genes. So this is a two peptome workflow based on the application select which one. Inside each workflow, we will use the algorithm based on the application. For each precursor, we try to see first, we do de novo and a database. Then if it's a database have a good match, we will go preference database peptide. Otherwise, we will check whether it's a modified peptide or it's a mutated peptide. And we take this preference. If all those was exhausted, we couldn't find one. If they have very good de novo peptide sequence, we will output as a novel peptides. So this is a workflow in all PICs functions, regardless it's DDA or DIA. So here I specifically introduce the homologous search for DIA, because DIA, the de novo is much complicated than DDA, because given any spectra, it generated from multiple precursors. Those fragment ions might cause de novo errors or interference. Another issue is DIA very often are very good for low abundant peptides, or considering now we search canonical database more than search those sequence variants database. And in that case, some of the database may not complete, can miss some information. Now, it's very important and necessary to take both information to get good true result of peptide sequence. Let's give a very simple example. See, there are some spectra, you may get this sequence. However, in the database is this sequence. At the beginning, you look at, they are significantly different, no common, except the last lysine. Actually, when we go detail the algorithm, we see if you segment it, the ARS and SAR, it have the same mass. It might, so de novo might have a mistake because of some tiny peak or miss some peak. It's assigned to ARS instead of SAR, and similar to FA and AF. However, we have a significant fragment ion to support in the third position is alanine instead of cysteine. In this case, the spider, the homologous search will come up with this sequence, which take our knowledge base, like say database information, and also show respect over mass spec information. Is in that position, we do observe the fragment line to support its anony is not assisted. So this algorithm, I think, compared to DDA, is more important for DI world. So in this release, we put this one into our DI analysis workflow, including Proton and Peptidone. And here is one example. For example, there is a data, and you take those one, you find a sequence. You find this is a very good de novo sequence. However, this spectrum cannot match to this sequence. There is a difference. Here is S, but here is T. And with this algorithm, you get will match with this one by convert this S to T, and we believe there is a substitution. And with that, surely this data set has RNA sequence. And from RNA sequence, we get the reference, and those two are exactly the same. Even they have 2 points changes, we still can find this. So this is just an example of how sequence variants can help us to find more peptide sourcing. And you may question how was this performance? And at this moment, we are very happy to tell you now we have a very good speed in terms of DI data. If you have GPU enhanced and just general graphics card, you can we guarantee that data analysis time is much less than data acquisition time. And for the sensitivity and accuracy, the algorithm was well controlled by target decoy or entrap database to control the data quality. And also this shows our algorithm have good sensitivity. And now I think I take 10 minutes around to show how we use the software to do data analysis. And here is our software. And take a look at this one. This is a software. If I think most of you familiar with PICS, technically for all the function button we put on top of this is like a function button or configure button. And here is a project. All the data loaded in and different data analysis, we archive them in this way. Here we have property to show each analysis. And this is the result pane. Okay, in this case, how to run those ones? Suppose you have a database configured. You can configure a database, enzyme, methodology for even your like say instrument setting. You can say in this case, in like my laptop doesn't have a GPU, this is not enabled, and you can allocate this one, and after you do those one, then you can do the data analysis. To do data analysis, click this button. Let's say I have a data, and here is my data, and I can use this one. Is to do this is a like say 2 condition data sets. Each condition we have 3 replicates run. They will have 2 groups. In this case, I can archive. You can use this regular expression button and to select those one past this. In this case, this is 1 condition. This is like we can use select this 2. Now you can see all data based on those give the name. You can clearly see this is diseased. It is Y type. And in this case, let's say if in this guy forgot it's DIA data, and you can go select DIA. Instead, you change all those one. You can select those apply to our project. Yet we will change all those one. Now we can select the workflow. In this case, because I try to find the protein which are significant change between these two groups. Obviously, I think a proteome workflow is my like a target workflow. So in this case, select this, and then we can go next. And in this case, we do match between one. Match between one is try to maximize the identification and like align those features come from the same peptide. To do this, now in this release is group based. So those are like diseased, and this is one group. This is another group. And see, now the match between them perform on those two groups. And after this, you can start, now we have auto detect for precursor and mass fragment ion setting. You just keep it as a default. And then we can do search. And you can do library search. Also, it's not a mass. You can skip over this. And then you can do direct database search. And so direct database search, you can set the modification. For example, this is the one we normally common for protein reduction and alkylation. And you can also select some modification. And for example, we select like say oxidation or deamidation. And very often we have those one. And okay, and then we can select trypsin digest, and it's a semi-trypsin or miscarriage, and you set all those parameter database. And this we are tell surgeon find the peptide in the database. If you check this one, it will find the sequence variant with this database to report. Or if you check this one, we also give you the lower peptides and by those settings. And then you can do, okay, next. And we can do label-free quantification. If you check a label-free quantification and take a look at the group, you can, based on your rules one, or you can regroup it. And then you set the parameters, and then you can go QC, or you want to quality control, or you can do a report. And for report now, and we have quality control, either protein FDR or peptide FDR. And let's say I focus on protein FDR or the score, and we also provide you an extra quality control. Precursor means not only at the protein level we want 1% FDR control. However, we also want at the precursor level is 1%. I needed to tell you that PIC's algorithm for precursor control is at a sample level. However, protein level is at a project level. That means we put all data together for protein control. Precursor control is for each LCMS run, we take control of precursor. If you select this way, is please control each data file, make sure the precursor FDR is 1%. With those one, also at the global project level, get protein FDR is 1%. Export those result for me. And this is a DRA selective. And in here, I show you one data set, which is from human. It's some in the ABF study, and they designed to like check how DRA compared to DDA and performance. And the first one, we take one DRA data, we do a database search. You can see after database search, we get feature, as I mentioned, and tab. And okay, it take a few seconds to get those one. You can see this is a feature, and it's a very low abundance feature. You can see, and the one you can see the peak matched is burying those high abundant. But you can see this is a precursor, and this is the fragment ion. You can see there are some fragment match of those, and if you thought about those, See, this is a higher bounded and peak. It will have much better like a fragment ion matched. So and DI, that's the one. And then we also have protein information. And the first summary will tell you those one. And look at this one. We are precursor based or feature based. And this is their identified and detected. And you can see one more yellow bar here. This shows the number of precursor peptides, which doesn't have very good detectable MS1 information. But this peptide provide enough evidence in the fragment ion such that you can use machine learning or deep learning information predict the precursor, the location, and find the like related fragment ion, get that peptide identified. And the number of those we are shown here. And so with those one, and say like you can see, we have protein tab, and let's say we have this protein tab, and this is a gray bar, shows there is a de novo peptides mapped to here. You can see, and from AR to Q exactly match, but some of those doesn't match very well, right? And this is the protein view. And one thing I want to show this one, this is a human data. HeLa data, I search human database. I also can search like a mouse database. If we look at the summary view, how many peptides we found, it's about 40 47,000 peptides. And if I search mouse, what are those peptides we found? You can see now significantly reduced. And now if you look at many of those, we are found in sequence variants. Look, if you look at here, this is a peptide, this is a DI, this is a variant. It shows that there is a mutation about those. If you see, this is their command. If you click here, it will show you where it is. See, this is a peptide, and it's a sequence variant. You can see there are two mutations together, and if we go the same precursor, go human database search, you will find this is exactly the sequence in human. And with this one, I would like to say roughly and show the idea about how pigs handle those DI data. Finally, this data sets I show you label-free result. And this is HeLa data sets. In this study, it was spiked some protein and which is spiked in the labeled as ABIF study protein. And those protein have different amount between those two groups. And now you can see if we go to protein view, and see, you can see here is the protein. Now, those are 4 protein. Show its ABIF 1, 2, 1, 3. You can see the 4 spike protein, there are significant change between the groups on the top. And if we want to say this protein, this is individual in each replica. They have 50 nanogram. Another one is 200 nanogram. You can see those are 3 replicates, and this is their ratio. Between replicates, they are very consistent. And then this is the final ratio. And you can see the theoretical one is 1 to 0.25. Now it's 0.28. I think it's pretty good, that like accuracy. With this one, I think I roughly introduced the PIX13 basic function. I didn't have a chance to talk about like a peptidome. Now I switch gear to introduced our another software which was designed for top-down proteomics. And we call this name is ProteoformX. That means there we try to find not just a protein, we try to find the exact proteoform. Based on current definition, where accepted definition is a proteoform is a base sequence plus the modifications and their positions. And that means the same sequence may have different and modification. If they have different position, then it's different proteoform. Or the major form is a major sequence is exact same, but some sequence they're missing. That's also another proteoform. So with this one, and I give the typical top-down mass spec workflow given any of those sample, and with protein mixture. Instead of like a bottom up, we use enzyme digest. Here we do denature or whatever native, and then we go mass spec. And what we get in the surface scale, we get is individual proteoform, their profile. If we deconvolute those one, we will get intact mass for each proteoform. And here, because surface scale, very often we get is a mass value, and it may contain different forms. Like this one, their mass is very simple, is exactly the same, but and they may have different positions about two modifications. If we are lucky in the retention time, they can separate. That's perfect. And then we will get tandem spectra, tandem events at different M/Z or retention time. If we are lucky, our tandem spectrum may contain those fragment ions related to those PDM positions. Then we can do identification at proteoform level. And this is an experimental workflow, and also follow this logic. You can expect PICS algorithm for proteoform top-down is proteoform feature-based solution. And take this as a very simple example. If we can contain those three proteoforms, and they're different with different modification. And in this case, and when we go mass spec in the surface scale, each proteoform will generate a series of precursor, which with different charges, because it's a larger molecule, it will have pretty bunch of precursors. And so how this algorithm will do it, we first do like a bottom up, do field detection. And then we group different features belong to the same proteoform and into one group we call proteoform feature. It's not a precursor feature, proteoform feature. And after this one, and then we will associate the tandem spectra with the proteoform feature. In this case, for example, those two spectra come from a different precursor of the same protein group. Yet those two spectra we are associated. They have slightly different fragment ion, but we are matched to the same sequence with different information. And then we will merge this to give the sequence information. Like this one, we are associated with the proteoform 2. And in this case, if our proteoform 1 doesn't have tandem spectra, and technically we have no way to identify this. Okay, this is the algorithm. It's proteoform feature-based solution, similar to our bottom-up. And based on those, we provide two workflow. First, we provide an intact mass analysis workflow, meaning given a protein mixture, just based on the intact mass, we tell you how many proteoforms are detectable at least. And this is one example from ADC. And see, you can see with this detect this mass, we have different group. These correspond different dot value, and within a group is different. It's a glycan profiling like a motif. And with this one, we can see as we expected, oh, this ADC is very complicated. They have different dot value, even though very often we measure the average dot value, make that, for example, is 3.4, but they have different proteoforms. Considering glycan profiling, they have very complex proteoform. And on the top, above the one is top-down search. If on top of those one, we have tandem spectra for some precursor with those proteoform. Here is one example, a proteoform we detect. Actually is FD parts, and in this case, there's 3 drug sites was identified based on those top-down fragment ion. Those are two functions we provide current in ProteoformX. And no surprising, the result presentation will follow the same logic we put in bottom up. For example, from feature perspective, you can see those are proton feature, and which this is an example of list antibody. And they have light chain, heavy chain. You can see this is a light chain protoform, and they have different charges. It will group together. There are some tandem spectra give you this sequence information. And if we put all those together, you can see we will have a protein and proteoform, and they are like a top-down information. And one more tool we put in ProteoformX is we provide graphics for you to define the proteoform you want to inspect. Here you call proteoform manager. With this one, you can load your sequence, and also you can pinpoint to site specific to set the modification. Either is PDM or the linkage and between different protein proteoform. And also you have glycan or disulfide bond, all those one. And after you select, you can have an overview to show okay different subunits how they connected together. This is an example of antibody. With this one, and this can let you easily define your interest of proteoform, and it's complex. With those one, yet those information will be used in the following workflow analysis. And up to now, I would take some time to show you how the software works. And here is the software, and similar to our PIX software, ProteoformX similar. Here is a project. You have many of configure buttons. And first, I introduce proteoform manager. Here you can determine any proteoform you want. And I just quickly show one of those. Hey, for example, here is a list antibody, and you have a heavy chain, light chain, and you can define those one. You can specify those, and then you can have an overview function to specify. Oh, in this case, they have N-terminal pyro group from Q or E, or they have a glycan, and they also have those linkage. And with those one, For glycan, it's in the heavy chain. If you click here, it show you those glycan forms. And this is one. Now you can, given a data, for example, you can do the data analysis and start from this, and you can load any of the data. And for example, I have the data. For example, I have a data here, and it's QTOF data, and I would like to do a CID, for example, and let's say I top down, and I like a CID, and then I can do top-down database search, and now you can specify. And here you can see we support first chimeric. And very often there is Windows, they have your target also chimeric. So it's similar to bottom up or top down, we also support a chimeric spectrum. And then you give the say the modification. Either you do fixed, then you set the modification, and then you do like a top-down database search. And in this case, and you can spec then you define the report, and for top-down score or Q value, or you can also choose I want the sequence coverage. This is one. And similar for bottom up intact, and you can do similar one. And now you can see I show you the result. First, I show you the intact. Here is the intact one. This is one HB95. It's a 3 antibody mixture. Even though many of us assume it's a monoclonal antibody, but it's an antibody mixture. And here you can see this is a whole antibody. We get 3 major peak. And if you do reduce, and we get heavy chain and light chain. See, you can see for light chain, you have 2 light chain, 1 heavy chain. And this is the one. And if we with here, the software will automatically do those assignment and put them together. And for example, here is a whole, and here is reduced. If you expand this one, this is from those 2 light chain, and this is another one, and this is another one, and we load them together. And the pairing software will automatically pairing. And if you here for each of those, I would introduce a little bit more. Here is the precursor belong to that light chain. Like I say, very often, proteoform have different charges, and we detect those one, and this can be exported. If we want to target the top down, this is a list for targeted top down. And this is the one. If you show with glycan, you can see, and the reduced one, now you can see they have different glycan forms. And yet now for whole, you can see now they have more forms about those 3 groups. And this is the one. Surely I can show you very complicated. It's ADC. Now you can see this is ADC, and we define it. We automatically do assignment when you select those. So, and this similar to those one. Now people are focusing the study of find site specific. If that's the case, and most of those abundant one, we would like to see only have 1 or 2 major form. Now, in this case, it's not the case. And finally, I show you one top down, and this is a list protein antibody. And we did this one, and I think you can see this is from protein, and we use like metadata, and you have FD, ARC, and FC. And for example, this is FC. We have many proteoform detect. If you want to see each of those, you right click, it will show in the proteoform table. And this is the spectra, and this is a coverage. In this case, okay, this is the coverage, and this is you mouse over it will tell you how this pick will come from. And if you go PRSM, here will show you this. And I think if we also if I show you the data, we have a data view here. And now if you go here, you have a jump, and then we can jump to LCMS view. And this one have a good point. You can see those are feature group which belong to this form. So those one, and if I raise this one a little bit higher, you can see those are protoforms belong to this FD, and there is a tandem spectra give the sequence. And with this data view, this is another one, and protoform, this is another one. And I think overall, this is a tool for ProteoformX for top-down proteomics. I think I stop here, and you are welcome to play with this tool.